Hey, hi everyone. Welcome to today's GCSE Biology lesson. We're just going to give everyone a couple of seconds to get in and get settled, make sure that everything's working okay. Okay, looks like there's quite a lot of you here now, so let's start. So welcome to My Tutor Online School GCSE Biology lesson today. This is a 1-hour lesson on stem cells. If you have been a part of biology lessons on the My Tutor Online School before, then you'll recognise me. My name's Charlotte, and I've taught all of the biology lessons so far. Today we're following on from yesterday's topic of growth and differentiation and cell division, and we're going on to talk about stem cells. So it follows on nicely from yesterday's lesson, but if you weren't in that, don't worry. Lots to learn and you'll be fine. You won't need to catch up. This lesson will be an hour long. We'll do about 45 minutes of content and 15 minutes of questions, but sometimes that spaces it out through the lessons. So there'll be a couple of, we've got some Slido word clouds today that we're going to do, and we've got some exam practice at the end as well. So if you look at the bottom of your screen, you've got chat and Q&A functions that you can press and open up. So for those, if you've got any questions about the content, pop them in the question and answer. And if you've got any kind of answers to questions that I ask or anything, pop them in the chat. And I'll make sure that I go through that question and answer section and answer your questions as we go along. Okay, so I'm Charlotte. I studied conservation and biodiversity at Exeter University. And now I work for a charity helping farms be environmentally friendly, which is great. Let's move on to today's lesson. So, as I said, last time we talked through surface area to volume ratios and how they relate to gas exchange surfaces, or just exchange surfaces. We talked about cell division by mitosis. And then at the end, we went briefly through growth and differentiation in cells, and we mentioned stem cells briefly there. This lesson, we're going to focus on stem cells and their uses in medicine, as well as we'll do a quick recap of what they are. And then we're going to go on and do some exam technique questions. So the last few lessons, we've worked through what would be module 1 of AQA GCSE Biology specification. But it's pretty much the same across all specifications. So if you're studying OCR when you're back at school, or WJEC, or any of the other exam boards, then don't worry, this will be relevant. Because we're reaching the end of that, I thought we'd do a bit of exam technique to kind of introduce you to the different sorts of questions that you might get in an exam. And then on Friday, we're going to work through and do a bit of revision as well. So let's do today. In fact, before we start, I wanted to do one of the Slido word clouds. So what I'm going to do is I'm going to put a link in the chat box in a second. So I'm just going to pop here. Okay, so in the chat box there is a link to a question. And if you click on that link, it will bring you to a screen where you can answer a question. And that question is, what are the keywords you can remember about stem cells? So don't worry if you've never studied them before, just put down what you think the keywords are about those. And we're going to kind of get an idea of your understanding so far. So this is really great. We've got loads so far. Differentiation and undifferentiated are the two key words. Well done, everyone. Cells, development, early stages, adult, bone marrow, well done. Loads of people have put bone marrow. Embryo, that's really good. Loads of stuff to learn about embryos and stem cells. Plants and animals, yes, they're in both stages. Roots and shoots we've got in there, brilliant. We talked about those yesterday. Specialized, exactly. That's another key word in this topic. Meristems, that's really good. That's for areas in plants where stem cells are produced. Replicators in there, that's really good. Transplants are in there, therapies, paralysis. Loads of cool things. Someone's got the textbook in front of them. Okay, cells, great. This is really fab. So this is a really good idea of lots of the keywords that we are going to use in this topic. So I think we'll stop that there. That's a really good word cloud there. Okay, well done, everyone. So thinking about all those keywords, we're going to crack on and have a look at some stem cell content. So well done, everyone, on that one. We're going to do another one of those at the end so we can see how that's changed. Okay, so what are stem cells and how are they used? So stem cells are undifferentiated cells that can divide to produce more undifferentiated cells. So last time we talked about differentiation of cells into specialized cells. So it's when cells go from a stem cell to turning their genes on and off, which allows them to differentiate into specialized cells that perform a particular function. So we talked about muscle cells which contract and relax and have special proteins for doing that. Well, those will have started as undifferentiated stem cells and then will have specialized or differentiated into those specialized cells with those proteins. Okay, so you can have, well, I suppose we talk about three different types of stem cells. So we talk about embryonic stem cells, adult stem cells, which are found in the bone marrow, and also meristems in plants. So we visited embryonic stem cells and meristems yesterday, briefly, but we didn't really talk too much about adult stem cells, which are found in the bone marrow. There's a nice diagram here of cell differentiation. So stem cells can become any different sort of cell. So blood cells, nerve cells, cardiac cells, liver cells, intestinal cells, any of the cells in our body. It depends. So different sorts of stem cells can have sort of limits to their differentiation. So embryonic stem cells can differentiate into any type of cell, whereas adult stem cells from the bone marrow can only differentiate into certain sorts. And yesterday we mentioned that red blood cells are one of those types of cell that adult stem cells can turn into. We also talked about meristems in plants and the stem cells that are formed there, which as a plant is continually growing, meristems produce stem cells and those cells gradually move through the plant as the plant grows and they become whatever cell is needed. So they may become a leaf cell, they may become a root cell, they may be a xylem or phloem cell, transporting things around the plant. It could be any of those. So plant cells are really, really adaptable. How do we use stem cells? So we've kind of talked about what they are a little bit, and we'll carry that on as we move through here. But because of their unique properties, their ability to turn into anything else, stem cells could be a really useful tool to us in science. So scientists would like to use stem cells to replace body parts or cells that are missing. So for example, they could potentially be used to replace nerve cells that have been damaged in spinal injuries, so to cure or go towards curing paralysis. In 2014, stem cells were successfully used in a trial to help improve the vision of people with seeing difficulties. So sometimes you can have a visual condition that blurs some of the image that you're seeing. So for example, you might have a blurry bit in the middle of your field of view. And stem cells were used to help kind of reduce that blurriness and improve the overall vision of people, which was really cool. So they've already been used quite successfully, but we're kind of at the stage where we're still testing for most things. So remember the part of a couple of weeks ago, I think we talked about developing drugs and how they go through a really rigorous testing process. Well, stem cells are kind of already in that process, but haven't made it out the other side yet. So they could also be used to produce insulin for people with diabetes. So type 1 diabetes is where your cells, you lack some of the cells that produce the insulin that the body needs to control the sugar levels in the blood. And we're hoping that stem cells could be used to generate those cells in people with diabetes. So replace what's missing. We also think that they might be able to be used to grow new organs for transplants. And they can also be used in horticulture. So we talked yesterday about taking cuttings of plants. And because plant cells can almost always change into any other type of cell, you can take a cutting or a piece of leaf tissue or any type of tissue of a plant. And with the right conditions, so be that in a laboratory, or perhaps if you've got a big enough cutting in some compost in a greenhouse, you can grow those on to make identical plants to the plant that you took the original piece of tissue from. So they will be genetically identical. So clones, we talked about yesterday. Plants are really easy to clone. In agriculture, it's a similar kind of use as well, mainly used in crops to make sure that you're producing, if you've got a really successful crop that grows really well on lots of different types of land, then you can use that to create genetically identical clones of that crop and use that for planting across fields to produce loads and loads of food. So stem cells, we think, could be really widely used. But there are some problems with using stem cells. So as I said, stem cell technology is very new. It's often still in the testing phase. And this means that there are some issues. So there are some issues with developing the methods needed, but there's also social and ethical issues. So this is a really kind of a discussion topic and something to really think about. So risks of using stem cells are that there's slow progress so far. So research has been quite slow. And a lot of time and money are being put into that, as you can see at the bottom there. So there's some questions about whether it's worth it, but most scientists think it probably is at the moment. There are some risks of viral infections and immune responses. So if you're using stem cells, you're putting, so particularly in humans, say, if you wanted to put stem cells in a body to produce those insulin producing cells that we talked about, or as transplants of organs, then it's possible that those stem cells can trigger viral infections and immune responses. So we did talk about viruses as pathogens, and viruses are very difficult to get rid of. and they, as we know at the moment, that's why we're all sat at home. So they move into cells, and then they replicate within a cell, and then they burst the cell. And it may be that stem cells are kind of more prone to those viral infections. They're not sure yet, but there is some evidence that they might be. Because of putting what your body might consider foreign cells in your body, even though they're human body cells, they're not from our human body necessarily, our immune response might be triggered. This differs between when you're using adult or embryonic stem cells. So there's much more of a risk of that immune response if you're using stem cells from another adult versus embryonic stem cells, which can turn into anything. There've also been some evidence that the use of stem cells might be linked with cancerous growths as well. So there's lots of these things to kind of bear in mind and think about when you're thinking about the use of stem cells in medicine. This is the sort of thing that in an exam would have a long question after it. 4 to 6 marks, discuss the use of stem cells. Social and ethical concerns. So embryonic stem cells come from embryos, and those are either embryos of babies that haven't gone to term, so stillborn births with the parents' permission could be used for that. Parents that have had abortions, they would also have embryos that could potentially be used as a source of stem cells, or people that have had treatment for fertility, so IVF treatment, may have what might be determined as spare embryos that again could be used as a source of stem cells. And lots of people have lots of different opinions about this. So if you're using an embryo, has it given permission? It can't, it's an embryo, but does it have a right to give permission for its cell to be used? Is it a good thing, a moral thing, an ethical thing to use the cells of a child or an embryo for that sort of thing? So there's lots and lots of ethical concerns there. They often tie in with religious beliefs as well about whether an embryo should be treated in that way. So there are lots of kind of social or ethical concerns, particularly around the use of embryonic stem cells. So for adult stem cells, which can be extracted from bone marrow from a consenting adult, there's less of a problem there, but there are still risks, and more risks in some cases. So there's lots to talk about there. Lots to think about for those longer questions in an exam. And it's a really interesting topic. Stem cell technology is very exciting, but it also has lots of issues. So the future. So what are we hoping to move towards with stem cells? So currently, stem cell research is hoping to treat the spinal cord after injuries, so by replacing those nerve cells that might have been damaged. Diabetes, so we mentioned the cells that are missing that produce the insulin, so hopefully stem cells might be able to replace those in a body. Heart transplants after a heart attack. Eyesight in the blind, we talked about that as well. There's already been that quite successful trial, although that wasn't on someone that was totally blind, but it was on people that had partial sight loss. And damaged bone and cartilage as well. Hopefully those might be treatable with stem cells, so producing those bone cells. Breakthroughs so far. So it's a nice diagram there that I'll work through in a minute, because I realise it hasn't transferred very well into my tutor, so I'll read through it. But breakthroughs so far are that embryonic cells have been found in umbilical cords and amniotic fluid of newborns instead of spare embryos. So there are a lot of words in that sentence. Embryonic cells have been found in the umbilical cord, so that's the cord that connects the embryo to the mother. and the amniotic fluid, which is fluid that surrounds the embryo in the placenta. And that's great because both of those things could be considered as kind of a byproduct of having a baby. You don't need them once the baby has been born. And so using those instead of spare embryos can combat some of those ethical or religious concerns that you might have. So secondly, we've managed to grow adult stem cells, and they have been used for heart disease and growing organs so far, which is very exciting. Again, this isn't common. There have been lots of kind of specific trials about this, and there have been some successes. But using those adult stem cells, we've managed to make that jump. So using those for treating heart disease and growing organs as well. Thirdly, something called therapeutic cloning. That's where cells from an adult produce an early embryo of themselves, which means that potentially new organs could be used without rejection. So let's talk through that. This is definitely an aspiration at the moment. It hasn't happened. But what they're hoping is that they can use stem cells from an adult. So, for example, those found in the bone marrow. But manipulate them so that they can produce an early embryo of themselves. That early embryo will have stem cells in it, embryonic stem cells that can turn into a much wider variety of cells than adult stem cells. And therefore, you could use them to create replicas, also potential new organs, but they'd be from your own body, so your body wouldn't reject them like it would do embryonic stem cells from another source. So therapeutic cloning is designed to get round that issue of rejection by the body. So we talked about the immune response that might reject those new cells. And the fourth point there, stem cells have been discovered in the connections between the liver, pancreas, and small intestine. So those are adult stem cells that aren't in the bone marrow. That's quite a new discovery. So between the liver, pancreas, and small intestine, and they can be used to produce those insulin-producing cells to control blood sugar. So it's those cells that are missing in those people with diabetes. That has worked in mice. So it's worked through kind of all the preclinical stages of testing. They've managed to test in mice, and the next stage will be going on to test in human volunteers to see whether there's any consequences or side effects of doing that. But you would hope that after testing on mice, which are also mammals, so have relatively similar body systems, that there would be a possibility of success in humans. So that diagram there, we've got an early human embryo at the top. You can see some of the stem cells at the bottom there. You can remove the stem cells from the early embryo, culture them so that they produce by mitosis, so they have the same genetic material in all of them, that same ability to differentiate into any sort of specialized cell. And the stem cells are then made to differentiate into different tissues. The examples that they've given there are spinal cord, heart, kidney, or insulin producing cells. And those could then be transplanted into a patient to cure them. We've kind of gone through that so far, and we talked about the risks of the body rejecting those cells that aren't from them, and the ethical concerns with them being from an embryo in the first place, risks in terms of viral infections or cancer, and the sheer time and money that goes into this research. But that's the idea in the diagram there. Okay, let's do some questions before we do the next Slido. So let's go through the question and answer. What level is stem cells in AQA year 10? This is right at the beginning of the specification, so it should build on a little bit from what you've learned in year 7, 8, and 9. and it will build on a few of the lessons that we've had before today. It kind of comes in the same section as parts of the cell, cell division by mitosis, and then we talk about this, how cells become different types of cells. Are we going to do a quiz? We're going to do a quiz on Friday. We're not doing a quiz today. We're doing Slidos today. What cells do scientists use to plant human ears on a mouse? That was a really famous example. The ethics of that, I think, could really be discussed. I don't actually know what type of cell they used for those. I assume an embryonic stem cell, they could then turn into an ear. Did 5G cause the coronavirus? I have no idea. Is the virus a pandemic? Yes. Was it made in a lab? I have no idea. Great stem cell questions, guys. Can you explain how a immune response becomes a risk to humans, please? Yes, that's a really good question. So when you have foreign, or what your body considers a foreign object or cell in your body, so that might be a pathogenic cell, so bacteria, virus, protists, or fungi. We talked a lot about those a couple of weeks ago when we went through that topic. Or, so for example, when you get a thorn in your hand, if you've been out in brambles, which I do quite a lot of, but I appreciate other people don't. If you have a thorn in your skin or even anything else, minor injuries, then your body immediately notices that that thing is foreign, that it isn't part of the body, and it sends out an immune response. So that immune response, as we've talked about in a few lessons a couple of weeks ago, and as you might have gone through in biology, that immune response involves white blood cells. And those white blood cells produce antibodies and try to clump together the foreign things so that other white blood cells can engulf and destroy them. That's the immune response. The trouble is, if your immune response gets too aggressive, which it might well do with other body cells, your immune system can get a bit overexcited, and it starts to deal with your own body cells as well as the foreign ones. So it gets a bit too thorough. And because of that, it can start to have an effect on your own body cells as well. And it's a bit more complicated than that. There's other things, like it sends a massive shot of adrenaline through you, and so it can have some hormonal effects, but also effects on other cells in your body as well. So it's quite complicated, but essentially that kind of overload of immune response starts to break down your own body's cells as well as those foreign ones, and you want to avoid that. So usually when a transplant takes place, they'll take immunosuppressant drugs to stop the immune system doing that until everything's calmed down a bit and it starts to hopefully recognize those foreign cells as part of the body. Do stem cells have a nucleus? Yes, they do. So stem cells are normal body cells. So they'll have all of those features of an animal cell or a plant cell. Oh, actually, that's a point. They do always have a nucleus. They always have a cytoplasm. They always have a cell membrane. Other parts of the cell that you find more of in specialized cells, they might not have to the same levels. So for example, cells that have lots of mitochondria or lots of chloroplasts in plants, stem cells might not have as many of those, but as they differentiate to become those specialized cells, they might develop more. If they have a nucleus, will they have different DNA if the patient uses others? Again, a really good question. Yes, slightly, but in the process of differentiating, cells turn on and off certain genes in those within their DNA, and that's what makes them specialized. So the turning on and off of the correct genes as the cell differentiates into what is needed in that body will mean that it will have very similar, if not the same, DNA as other cells in the body that are doing that same job. So, for example, if you were talking about adding in extra nerve cells for paralysis to replace those ones that have been damaged, they would turn into nerve cells, which are pretty much the same as all the other ones in the body. So yes, you're right, they'll have a different kind of genetic source, but for kind of body parts, that shouldn't, it would still be able to perform the same function. Can stem cells help with reproduction? That's a really good question. I haven't heard of them being used in that way. Because even when you're undergoing fertility treatment, as far as I know, and as far as it goes in the GCSE spec, that fertility treatment is from spare sperm and eggs, which are used to produce embryos. So they're not from stem cells from embryos to produce a human being. So that's really interesting. And I don't really know, but I don't think so. Okay. Oh gosh, there's loads in the chat as well. Don't think there's anyone here that we're not going to answer later. Why don't they get the stem cells from the patient so it's less likely than your body rejects it? That's a really good question. Let's answer that one. That's from Alina. And the answer to that, which I may have been made clear, I don't quite know when that question was asked, but the answer to that is that stem cells from a patient, so adult stem cells, can't turn into as many things as embryonic stem cells. So that's why sometimes it's a real disadvantage to use adult stem cells. But you're right, if you use stem cells from your own body, there's not going to be that rejection risk. Do immunosuppressants prevent immune systems or immune responses from being as effective as they could be? Yes. If you're taking immunosuppressant drugs, you have to be really careful. You're much more susceptible to lots of other types of disease as well. So you're more likely to get colds, bacterial, viral infections if you're on immunosuppressants. So worth it in the short term, but you have to be really careful when you're taking them. When they differentiate into red blood cells, do they lose a nucleus? Yes, because red blood cells do have a nucleus. And exons and introns, we've got another question of there on the chat. They're not really relevant today, but exons and introns are different sections of DNA. So they're described as non-coding and coding sections of DNA. So some sections of DNA contribute to the different proteins that are used in the body, and other parts of the DNA aren't. So that's what an exon and an intron is. Okay. So now we're going to, because that was kind of the last bit of this module. So we're going to do another Slido quiz quickly, see if we've got any differences. So I'm going to post this into the chat again. There we go. Click on that one. Pop your answers to the questions. What are the key things to remember about stem cells? Here we go. Let's see how that's different. So remember in the first one, we had lots of cells, differentiation, things like that. Oh, good, we've got a joker. If anyone wants to put good, can cause cancer. Embryonic and adult cells, brilliant. Yeah. Any others? Umbilical bone marrow, yes, brilliant. Babies. Time and money spent on the research, that's really good. We have adult or embryonic, we've had that one. Spinal injuries. Regeneration. I wouldn't say that reproduction is a key thing to remember about stem cells, but someone really likes typing that in. Probably the same person that's been asking me repeatedly whether we can have a quiz. Mice, good. Range tissues, bone, viral infections, brilliant. Repairing things, yes, that's a really good one. Treatments, testing, good. Need to remember that about stem cells, that they are always in a testing phase. Slow progress is in there as well, that's really good. Fab, so this looks completely different from what we had earlier. There's a lot of the same keywords, so we've remembered a lot, but there's also lots of new ones in there as well. Great. That looks really good. Okay, so I've stopped that one. And now let's head back to, okay, back to our slides. Thanks everyone, that's great. So now we're going to do some exam technique questions. So I wanted to run through kind of how questions about stem cells would look in an exam, and also the different types of question that you can have, and we'll run through the mark schemes for those as well, so you can see exactly how those would appear, and hopefully you'll get an idea of what the examiner is looking for. That's the idea. So first one here, the first thing that we always do is underline the question, or the keywords in the question. So here we've got some really simple 1-mark questions. 1 mark for both. So stem cells can be taken from human embryos. In therapeutic cloning, remember we talked about that, an embryo is produced that has the same genes as the patient. That helps with the issue of rejection that we talked about. So someone asked that really good question about why adult stem cells would be more useful. Therapeutic cloning hopefully gets around that problem. So the first one says name one source of human stem cells other than human embryos. And then part 2 says that stem cells from embryos can be transplanted into patients for medical treatment. Give one advantage of using stem cells from embryos compared with stem cells from the source you named in part 1. 1 mark for that. Okay, so on the next page, I've got the mark scheme. So for part 1, which was name one source other than human embryos, you could have had bone marrow. That tends to be the one that everyone remembers. But they would also have accepted umbilical cord, skin, amniotic fluid, or membrane for those. So this is what the mark scheme looks like for that. Lots of different alternatives, but generally it'll be bone marrow. That's the most common one that comes up. And then for part 2, remember that's 1 mark again. So that was give one advantage of using stem cells from embryos compared with cells from the source you named in part 1. So say that's bone marrow. Why is it an advantage of using stem cells from embryos compared with bone marrow? And as we talked about, you could have that cells would not be rejected by the patient's body if they've been produced by therapeutic cloning. Allow easier to obtain linked to embryo stem cells. Embryo stem cells can develop into many different types of cells. That's the major advantage. Or embryo stem cells are not yet differentiated or specialized. So lots of different options there, which is good to know because they're not looking for one particular one. You can have any reasonable answer for those. They've outlined the most common there. Another short answer question. So we've got a nice bit of information here, a good table. So stem cells can be collected from human embryos and from adult bone marrow. Stem cells can develop into different types of cells. Okay. The table gives information about using these two types of stem cell to treat patients. So really nice summary table here. So stem cells from human embryos, it costs £5,000 to collect a few cells, whereas stem cells from adult bone marrow, it costs £1,000 to collect many cells. So bone marrow is slightly cheaper. If there are ethical issues in using embryo stem cells, whereas adults can give permission for their own bone marrow to be collected. Stem cells can develop into most other types of cell when they're from an embryo, whereas stem cells from bone marrow can develop into only a few types of cell. Answered that one on the previous question. Each stem cell divides every 30 minutes for embryos, and each stem cell divides every 4 hours for bone marrow, so they're a lot slower with their dividing. There's a low chance of a patient's immune system rejecting the cells if they're from an embryo, whereas from bone marrow, there's a high chance of patient's immune system rejecting the cells. More research is needed into the use of embryonic stem cells because we're not quite sure how they work yet, whereas adult bone marrow has been used for quite a few things and is considered safe for some things. Okay. Scientists are planning a new way of treating a disease using stem cells. Use the information above to answer these questions. So A was give 3 advantages of using stem cells from embryos instead of from adult bone marrow. So think about what we've just read and think of 3 advantages, and we'll move on to the mark scheme on the next slide. And part B, give 3 advantages of using stem cells from adult bone marrow instead of from embryos. So 3 marks for each. And they're slightly odd questions because they're asking the same thing but the other way around. So they want 3 good things about stem cells in the first one from embryos, and 3 good things about stem cells from bone marrow for part B. So, for part A, 3 advantages of using stem cells from embryos. 3 marks, so you need 3 points. Comparisons aren't required but should be credited, except a clear indication of the statement, even if incomplete. So you can see in the mark scheme there, there's some leeway. So I always think it's quite reassuring to look at these. So, can develop into most other types of cell. Embryonic stem cells, there are a wider variety of things that they can turn into, specialize, differentiate, and specialize. Each cell divides every 30 minutes, so they're faster, and a low chance of rejection by the patient's immune system for embryo stem cells. And then for part B, 3 advantages of using stem cells from bone marrow. Again, any 3 from the variety here. It's cheaper, only costs £1,000 only. You can collect lots of them at once. Adults can give permission for their own bone marrow to be collected, and they're much safer to use because we know more about them. So any 3 from that list would give you the 3 marks there. Okay, those are quite nice 3-mark questions, actually. So really, that question, rather than your own knowledge, was testing your ability to read a table, which is why it's really important to read it correctly. So things like that, that was 6 marks that you could get by just looking at that table. So it's really important to read the question properly and make sure that you're quoting the right thing. It would be a shame to lose a mark there because you hadn't quite read the question properly or the table. long answer questions. So we'll run through this one. I think this is the last one that I've got. And then we'll do some more questions. So, got a passage here about stem cells. So stem cells are used to treat some human diseases. Oh, terrible underlining. Stem cells can be collected from early embryos. These stem cells have not begun to differentiate, so they could be used to produce any kind of cell, tissue, or organ. The use of embryonic stem cells to treat human diseases is new, and for some diseases, trials on patients are happening now. Stem cells can also be collected from adult bone marrow. The operation is simple but may be painful. Stem cells in bone marrow mainly differentiate to form blood cells. We talked about that earlier on when we talked about how they're often used for red blood cells. These stem cells have been used successfully for many years to treat some kinds of blood disease. Recently, there have been trials of other types of stem cell from bone marrow. These stem cells are used to treat diseases such as heart disease. So nice bit of information there about how bone marrow is used. For 5 marks, evaluate the use of stem cells from embryos or from adult bone marrow for treating human diseases. You should give a conclusion to your evaluation. Evaluate is a command word that we don't see very much. It's not quite explain, and it's definitely not state. It's asking for your opinion. It's asking for you to evaluate some evidence. So this is moving towards scientific thinking. And they want you to give a conclusion. So this is a mini essay. So let's have a look at how they mark that. So this is a copy of the mark scheme there. They've been very quick to state, maybe do this with a highlighter instead. Marks should not be awarded for simply copying the information provided. They said evaluate, they want you to do your own thinking. A mark may be awarded for a comparison between treatments if the answer only involves copied information. So the only time that you would be allowed to copy is if you're making a comparison between the 2 different types of stem cell and how they're used, because that shows, your initiative in making that comparison between them. You can have 4 marks, and for all 4 marks to be awarded, there must be 1 pro and 1 con. The final mark is for your conclusion. They specifically said that you needed to give a conclusion, so they're giving a mark for it. Any conclusion. Well, a reasoned conclusion from the evidence. They've qualified that at the bottom there. So for embryo stem cells, they've got some examples of the pros. So you can use embryonic stem cells to treat a wide variety of diseases. They are generally quite available. Using them, it might be said, is better than wasting them, although there are some ethical concerns there. And it's painless to collect them. Cons of embryo stem cells is that there's possible harm or death to an embryo because you're using that embryo for stem cells rather than allowing it to develop into a human being. It's relatively untested so far, so it's still in the development process. And the embryo can't be asked, it can't give its permission. So that idea of an ethical problem there. If you're talking about bone marrow cells, pros of using those are that there's no ethical issues, or permission can be collected from the patient. So you haven't got that problem there. Relatively quick recovery, and they're relatively safe. And they're well tried and tested for treating those blood diseases. Cons are that you've got the hazards of an operation to extract those bone marrow cells. So there's all the risks of that, of infection. There's relatively few types of cell that can be produced. So we said that it's mainly blood cells, but they have moved towards heart disease, they said in that passage. And it's painful, so it may deter some donors. And all of those points you can deduce from that passage that they gave you. They've been quite kind here. They could have just said, evaluate stem cells from embryos or bone marrows, but they have given you a passage to work from. So you needed to have at least one pro and one con, and 4 of those points. And finally, you need a reasoned conclusion from the evidence. So anything that follows on well from the points that you've made. So I have done model answer here. So this would be a 5-mark answer. It's not particularly long, as you can see. I've tried to give 4 points and a conclusion. So let's move back to a pen. I can have a look at the points as we go along. So embryo stem cells can treat a wide variety of diseases. However, they do have ethical concerns, as an embryo cannot give permission for cells to be used. There's probably 2 there. Adult stem cells do not have these ethical issues, however, can only differentiate into relatively few types of cells, so their use is limited to few diseases. Another 2. Therefore, it is better to use adult stem cells and only use embryonic stem cells when there is no alternative. So that was my answer there. For this question, you could have concentrated on one or the other, so embryonic stem cells or adult stem cells, and any reasoned conclusion from those you could have given as well. Okay. So that's the content that we have for today. I'm going to have a look at some more questions now. Okay. Someone said for 6 marks, would you just give an extra point? If you've got a 6-mark question, then yes, but that question was for 5 marks. So the maximum you could have got was for 5. Someone's asked, can we explain therapeutic cloning again? Yes, no problem. Let me move back to this slide. So therapeutic cloning is when cells from an adult are used to produce an early embryo of themselves. So cells from the patient are used to produce embryo, or to produce, embryonic cells. That means that it's from your own DNA. Though that embryo could then be used as a source of embryonic stem cells, which could be used to create organs or anything else that might be needed and inserted into the patient without rejection, because the origin would be from the person themselves. That's therapeutic cloning. Oops, someone's just said that I'm frozen. I think all is well. Paula, let me know if I'm freezing for you, otherwise it might be an internet problem on the other end. Okay. If a person's stem cell can change the DNA of another's offspring, I'm not quite sure what that question means. You might want to pop that one on again. What is the scientific term for a singular strand of DNA in a single cell? We went over this a couple of lessons ago. It's a prokaryotic cell rather than a eukaryotic cell, which is animals and plants. Oh, more people saying that I'm freezing. Okay. I think we're okay. What's an umbilical cord? An umbilical cord is a cord that attaches a baby from their belly button to the placenta in the mother. So the placenta is kind of a sac that surrounds the baby, and it's how the baby gets nutrients from its mother. So the nutrients goes from the mother's body through the umbilical cord and straight into the baby. Wouldn't unused stem cells from IVF be thrown away anyway? Yes, that's a really good point. They're spare embryos. They weren't going to be used, so might as well use them. But again, if you believe that an embryo should be able to give permission, then I think you would still have some concerns with that. Are stem cells found in plant cells? Yes, they are. So stem cells are found in the meristems of plant cells. Hold on, I'll get up yesterday's slide and we can pop on the slides about that. I'll show you the diagram. So yes, plant cells have stem cells as well. And we kind of touched on this at the start, but plant stem cells can turn into anything. And throughout their kind of lives as a cell, they can turn into different things as well. So a stem cell can be produced at the meristem of the root. And as the plant grows, it can move through the plant and it could become a leaf cell. Or it can move downwards and it could become a root hair cell. This is just loading up now, forever, as usual. Always takes forever when you need it to be quick. So if we move to plant cells, okay. Let's pop this in the new page. There we go. So this is the slide that we looked at yesterday for differentiation in plant cells. So this diagram here is of a root meristem. So this is at the bottom of a root. They have this funny section here where it's called a meristem, and it's where stem cells are produced. So those stem cells are produced here, and then they slowly make their way up the root. We've got another one here at the top of the stem as well. They're really odd looking, but that's an area where stem cells are produced, and then they move around the plant from there. Okay. Just running through any more questions. Are meristems a big part of the GCSE? They are a key word. They do come up in all of the different syllabuses, so it's worth knowing about them. But all you need to know is that they're where stem cells are produced in plants. You don't need to know any more detail than that. Yeah, someone's put that embryonic stem cells may lead to new, more effective treatments for serious human ailments and alleviate the suffering of thousands of people. That's completely true. Looks like I've frozen for a few people. I'm sorry about that. Do I think a vaccine for COVID will ever be made? They're working to develop one now. So fingers crossed, yes. There wasn't ever a vaccine for some other diseases, that's true, but they are planning on doing one for coronavirus if they can. But who knows? None of us know. Okay. Perfect. I think that's all of the questions. So that is it for today. So on Friday, we're going to go through some of the maths parts of biology. So we're going to have a look at microscopes and the different calculations for them, how to convert between different types of units. And we're also going to do a bit of revision about all of this topic. So if you have any questions about parts of the cell, about cell division or stem cells, or different types of transport in cells, then bring those on Friday. And we'll do a quiz as well, I think, on Friday. Okay, hopefully see you then. Remember to keep checking into your timetable on My Tutor Online School, and have a great week. Thanks, everyone.